Hello guys, welcome to my YouTube channel. So today we will start the first type of vaccine, which is called as a live attenuated vaccine. So first is, what is a live attenuated vaccine? So it is a vaccine that contains living microorganism living microorganism that has been weakened, okay, weakened or attenuated under laboratory conditions. So what is the purpose? Why has it been done? It is done so that it loses its ability to cause severe disease, but retains its ability to stimulate an immune response. So this is very important, that it is a vaccine which contains living organism, but what kind of living organism? That living organism is weakened or attenuated under laboratory conditions. So the organism has been weakened by external agencies and it has been done in the lab, and the purpose is so that it loses its ability to cause severe disease, but at the same time it retains its ability to stimulate an immune response. Okay, so as you know, the word attenuation, if we talk about the word attenuation, it means in immunology or pharmaceutical biotechnology, it means reducing the virulence, okay, or disease causing capacity of a pathogen without eliminating its antigenic properties. Okay, so you reduce the virulence, but you do not fully eliminate its antigenic properties. Okay, so in other words the pathogen remains alive but becomes less dangerous. So this is what it means. So this live attenuated vaccine generally mimics a natural infection, and it allows the immune system to generate a broad and durable immune response. Okay, so this is the basic background of live attenuated vaccine. Now we will start with a bit of the historical part of the live attenuated vaccine. So if we talk about the historical part, history, okay, so the concept of attenuation, so this concept of attenuation was pioneered by the French scientist Louis Pasteur. So during the 19th century, so during this 19th century this concept was developed. So Pasteur discovered that if microorganisms were grown under unfavorable conditions, so what happens? They gradually lost their ability to cause the disease. So, however, these weakened pathogens could still induce protective immunity. So, his work led to the development of vaccines, attenuated vaccines against chickenpox, anthrax, and rabies. So, today, this attenuation is one of the most important strategies in vaccine development. Okay, so you can ask, so how are these live attenuated vaccines produced? Okay, so if we talk about that, so the main objective here is reduce the pathogenic virulence. So what is the main objective? Main objective here is reduce pathogenic virulence but preserve its antigenicity. Okay, while you are reducing the pathogenic virulence, you have to preserve its antigenicity. So there are several methods. So first method is serial passage in non-human cells. So what is done here? The pathogen is repeatedly cultured in cells or animals that are not its natural host. So as you culture the pathogen in continuously in those cells or animals which are not its natural host, what happens? It gradually loses its virulence. Okay, so for example, if we talk about human viruses, may be grown in chick embryo cells or monkey kidney cells. Okay, so you are using mammalian cells because humans are mammals in this way, but different mammal. So in this way you are preserving its antigenicity, but because the pathogen is specific to humans, it won't affect the chick or the monkey kidney cells, so the reduction of the virulence happens. So over multiple generations, the pathogen adapts to new environment. So during this adaptation, mutations accumulate that reduce its ability to infect humans effectively. So now you know why it loses its virulence. Okay, so this process is called as the serial passaging. Okay, now we come to the second type, which is the temperature using temperature sensitive mutants. So scientists may select viral strains that grow efficiently at lower temperature but poorly at normal body temperature. So as a result, what happens? The replication of the virus inside the human body is limited. So replication of virus within the body becomes limited. And number 3, which is the most common, is by genetic engineering. Okay, so this allows targeted modification or deletion of genes associated with virulence. So first you identify those genes which are associated with virulence, then you delete those genes. So in this case, in this way also the virus loses its virulence. Okay, so that's it for today's video. Stay tuned for more upcoming videos on live attenuated viruses. Thank you.